Hi everyone, I am Rajendra Surawase, Assistant Professor at Lokneti Dr. J.D. Power College of Pharmacy, Kalwan. Today we are going to discuss the lyophilization basics. So in this chapter, we will discuss the introduction to lyophilization process, the principal steps involved in lyophilization process, the different application of freeze dryer process, various components, parameter of freeze dryer, construction and working of lyophilizer or freeze dryer, and different excipient or ingredient which is used in a formulation of lyophilized products. Discuss the introduction to lyophilization process or freeze dryer process. So before the introduction of lyophilization process, we need to know about the rationale behind the formulation of lyophilized product. What's the rationale or what is actually need to develop a lyophilized product? Objectives of lyophilized product. So very important objective or need to develop such a type of or lyophilized product is stability. Most of the biological products such as the proteins and peptides and many antibiotics are unstable in a liquid or unstable in a solution and to be available as a liquid dosage form. Okay, so this is the very important reasons or rationale behind the development of lyophilized product, because the many pharmaceuticals and biological products such as the proteins and peptides are unstable in a liquid to be available as a liquid dosage form. So therefore, there is a need to formulate such a means proteins, peptide, biological products, antibiotics in a solid dosage form, solid powders. For example, lyophilized product or freeze dried product. So therefore, such a formulation can be filled as a solution and freeze dried under a low pressure to remove the solvent. Definitely, for the formulation of freeze dried product, we are going to use different solvents. So under a pressure, under vacuum, we need to evaporate or remove this solvent, and resulting in a dry formulation which has a long stability. So the proteins, peptide and many biological preparation must be freeze dried for storage and the stability. So this is the rationale behind the development of lyophilized or any freeze dried the product, stability. So therefore, see, under the pre-formulation study, why need to conduct the stability, means physical stability, then chemical, solid state stability, liquid state stability of a drug. Okay, so that is the reason. Okay, suppose we found the stability of that particular drug or biological product in a solution form, means unstable in a solution form. So there is no any sense to formulate such a type of a drug in a liquid dosage form. We have to develop or to avoid or prevent the degradation of a drug from these mechanisms. Okay, so the lyophilization, see the definition of lyophilization is a stabilizing process. Stabilizing process means to stabilize the drug molecule, biological product. So in which the substance is first frozen and then the quantity of its solvent. The quantity of its solvent is reduced or reduced is first by the sublimation. And this sublimation is also called as the first drying or primary drying in lyophilization process, sublimation. And then the desorption, that means the secondary drying. Okay, means the primary drying means the sublimation, and secondary drying the desorption. So these are the very important steps involving in a lyophilization process. Moreover, the lyophilization and freeze drying are terms that are used interchangeably depending on the industry and the location where the drying is taking place. Controlled freeze drying keeps the product temperature low enough during the process to avoid the changes in the dry product appearance and the characteristics. The structure, the chemical structure of the drug molecule. Okay, so this is again very important. Unchanged form of API, unchanged form of biological product after the formation of the lyophilized product. So this is about the, in brief, the introduction, the rationale, the various objectives of the lyophilization process or lyophilized product, also called as the reconstituable product. Okay, means there is the need to dilute or reconstitute this product before the injection, before the administration via any suitable parenteral route of administration. For example, intramuscular, intravenous or subcutaneous. We need to dilute or we need to reconstitute with the sterile water for injection or water for injection. So this time is also called as the reconstitution time. Okay, and this is very minimum, means the time required to form a clear solution or clear suspension. This time is known as the reconstitution time. Okay, now see the advantages and disadvantages of this process. So first advantage, the freeze drying process improve the storage life and marketing of the end product. Marketing of the end product of particular biological or proteins and peptide and antibiotics. So definitely the freeze drying process improve the storage life, the storage of a drug or storage of this particular compound in a solid state or in a freeze dried state. Second advantage is it's remove the water or moisture without the excessive heating. Without excessive heating. See what will happen in the case of excessive heating, the chances of the degradation of API as well as the excipients will happen. So it removes the water or the moisture or the solvent without the excessive heating, means under reduced pressure. Okay, and enhance the stability of a product in a dry state. So this is the second advantage. The next advantage is the distribution of components within the drying, means the primary and secondary drying, and dry products remain the unchanged without means throughout the process, means unchanged means the structure of API will unchanged throughout the process. The next advantage is the product is sealed under the vacuum or in the inert gases. So the oxidative denaturation or chemical decomposition is minimized. Next is there is no concentration of the solution prior to drying, hence the salts are not responsible to denature the proteins. Next advantage is the transport cost of product is minimized due to the loss of product weight in the dry form. The transportation, the cost due to its means loss of product weight in a dry form. I would like to tell you one story or one a case study of one process which is widely used in the pharmaceutical industry for the preparation of co-processed excipients or co-processed the polymers. Means the combination of one or more excipients in a single form or in a single mixture, and the single mixture you can use as a blend, powder blend for the preparation of the different, means conventional, whether suppose you want to develop or formulate the conventional tablets, so you can use the particular the powder blend, means co-processed blend. Either you want to develop a sustained release powder, so you can use the co-process sustained release co-process powder blend for the preparation of that particular the dosage form. So means how to reduce the transportation cost to this co-processing of excipients. So definitely after World War II, I think that is a huge demands of milk or the food ingredients, and it's quite difficult to transport the food ingredient or milk from one place to the another place. So in that cases, the introduce this spray drying technology for the production of milk powder. So definitely the transportation cost of product is minimized due to its the weight, okay, the loss of product weight in the dry form. And then after World War II, this spray dry technology or spray drying equipment is widely used in food industry, then pharmaceutical industry, and there are many applications of spray drying technology, okay. So that is what similarly in a freeze drying process, the transportation cost of product is minimized due to the loss of product weight, okay. Then the next advantage is a suitable technique for the preservation of a temperature sensitive product such as the enzymes, then blood product, vaccine, microbial cell, etc., etc., okay. For example, a fractionation of blood, preservation of RBCs, preservation of platelets, preservation of the blood components, preservation of enzymes, okay, vaccines. So this freeze dry technology is widely used and definitely suitable technique for the preservation of these highly temperature sensitive the components, okay. Then the lyophilization operates in a controlled environment, hence the products are free from particulate matter or contamination, okay. Control environment means control temperature, control pressure, control air pressure, okay. So therefore the chances of the particulate matter or contamination is a minimum in this process. So these are the some advantages, or you can say the benefits of the formulation of these products. Let us see, it has some disadvantages. The first disadvantage is this process required the more handling and processing time, as well as the cost of equipment is required for freezing drying is high as compared to the vacuum oven, okay, as compared to the conventional technique, as compared to the solvent evaporation, that means the old method or conventional method. So this technique is costly, so definitely the product cost will be higher than the conventional method. So this is the one disadvantage, or you can say the limitation of this process. Lyophilization is less suitable for a material which supercool to form a glassy state, and for certain cells which are unable to retain the viability, okay. And the third disadvantage or limitation of this process is the product formed by this process is hygroscopic in nature, hence the packaging requires the special condition. So these are the important steps involved in a lyophilization process, such as the sample preparation, freezing, primary drying, secondary drying, and formation of final product. So let's discuss one by one steps in brief. The first step is a sample preparation or a pre-treatment. See, the pre-treatment or formulation is the key method or a process of treating the product prior to freezing, and the processing and formulation development are important steps often taken to make a product ready for freeze drying and usable for its specific application. Means uh, before start the actual process of a lyophilization, we need to know about the all the physical, chemical, microbiological product characteristics, properties of drug. Means suppose you are going to formulate the biological product or any antibiotics in lyophilizer product, so we need to study thoroughly all the physicochemical properties of that particular API and all excipient prior to the freezing stage, prior to start the actual freezing. Then the selection of a excipient for the formulation can affect the thermal characteristics of the product and drying time. So therefore, under the pre-formulation, we need to study its a differential means thermal stability study, thermal study by using the DSC analysis, differential scanning calorimetry analysis, TGA, FTR, etc., etc. So this is about the sample preparation and the pre-treatment of a sample before a freezing. So next step is the freezing. So freezing is a critical step in the freeze drying process because this stage form microstructure porous product and it determines the quality of the final product and processing characteristics. Okay, means formation of a microstructure of a product or formation of means porous products in these freezing steps. So therefore this is the very critical steps in the freeze drying process. Okay, and the principal functions of this freezing process is to separate the solvent from the solutes and minimize the thermal degradation within the dispensed product and to prevent the product foaming when vacuum is applied. Okay, so this is about in brief the freezing steps. So now see the primary drying. After pre-freezing of the product, ice can be removed uh, from the frozen product via sublimation and to form a dry intact product. And this primary drying, also called as the sublimation, is a slow process conducted at a cooler temperature safely below the product critical collapse temperature. Okay, and the rate of sublimation of ice from the frozen product depend upon the differences in vapor pressure of the product compared to the vapor pressure of the ice collector. So this is about the primary drying stage. Next step is the secondary drying, also called as the desorptions. The primary drying process leaves the product with about 5 to 10% moisture in the solid, and this is removed in the secondary drying process. Okay, so in many cases such a moisture means 5 to 10% moisture remains after primary drying, and this 5 to 10% moisture is removed in the secondary drying stage, means a desorption through a desorption. Okay, in many cases such moisture values may be too high and the final product may not have the desired stability. Suppose after primary drying this moisture level is means about 10%, about 20%, so definitely 20%, 25% is not completely removed in a secondary drying process, and so therefore its influences on the final product characteristics. So therefore this stage is very important. Secondary drying process start during the primary phase but at elevated temperature in the range of 30 degrees Celsius to 50 degrees Celsius. Okay, so this is about the secondary drying stage. And after a secondary drying, so you can collect the sample in solid state, highly porous, highly microstructured lyophilized product and pack in a suitable container. Generally vial is used for the packaging of lyophilized the product. So this is about the in brief introduction to various steps involved in a lyophilization process. So this is about the introduction to the lyophilization process with different advantages, disadvantages or limitations, and some important steps involved in lyophilization process. In next video, we will discuss the principle of freeze drying, the various applications, construction and working of freeze dryer, and the different excipients and polymer which is used in the formulation of freeze dry the product. Thank you very much for watching this video.